Welcome back, everybody. I hope that we are ready to continue on with our exciting lecture on cellular movement and muscle. So last that we left off, we were talking about the sliding filament theory, and we were talking about the fact that your muscles have actin and myosin, the thin and the thick filament, and they need to go ahead and make physical contact or cross bridge. And they need to move or slide past each other, which is the formation of power stroking. And that all correlates to the fact that we can see our muscles contract and relax. Now, I wanted to show you these pictures right here because these are some histology pictures of striated and non-striated muscle. And hopefully some of you have seen these pictures before, right, in previous classes that you have taken. And if you haven't, it's not a big deal. But basically what I want to show you is from a histology point of view, when we look at muscles, we see that they're either divided as, or they're either classified as striated or non-striated. Striated means that when I look at my muscle fibers, so these are my individual muscle fibers, what I'll see is I'll see these perpendicular lines running in a very organized format. Non-striated means that I'll still see my muscle fibers, but there are no perpendicular lines running through them. And it turns out that the striations that we're seeing, those are the actin and the myosin lined up perfectly together. So you have a thick filament followed by a thin filament, then another thick, then another thin, very organized, bundled together, and this is then what creates the striated version. So you're looking at actin and myosin. On the other hand, non-striated muscle will still have actin and myosin, but they are very abstract in their shape. They're kind of arranged all over the place. They are not bundled in parallel, so therefore the striations do not show up. Now, there are three types of muscles in our body. We have skeletal, cardiac, and smooth muscle. Skeletal and cardiac muscle both fall in the striated category, whereas smooth muscle is the only non-striated muscle that you're looking at. And smooth muscle is usually the one associated with your internal organs. So as you can see, you have the viscera, are your gland secretions. Part of your blood vessels will be smooth muscle as well, whereas your skeletal muscles, we usually associate that with the skeletal system, so the movement of the arms and the legs, the facial muscles that allow for facial expressions. And then the cardiac muscle, that one's going to be the unique one to the heart. So a striated muscle cell, now that we know that it's made out of organized pattern of thin and thick filaments or actin and myosin, we could also talk about the fact that from a structural unit, the smallest part that's able to contract is going to be called a sarcomere. The sarcomere is the contractile unit of a myocyte, and a myocyte is a muscle cell. And what we see happening is within the sarcomere, you'll have your arrangements of your thick and your thin filament, and you'll also have different indications. So what I want to do is I want to talk to you about the different terminology, and then in a few slides we'll take a look at a picture of a zoomed in muscle so we can see all these different regions that I'm going to be mentioning to you. So first things first, the smallest part of a muscle that can contract is going to be the sarcomere. That is the functioning contractile unit of the muscle. A sarcomere is composed of thick and thin filament, so your myosin and your actin in parallel format. And what we see happening is that the borders of the sarcomere are represented by Z discs. So from one Z disc to the next, we'll be representing a sarcomere. We will also see that when we go ahead and look through the muscles with an electron microscope, we're going to notice the fact that they have what we like to call an A band and an I band region. The A band region will be indicating the fact that you have lots of thick filaments in that particular area, creating more of a darkened format, and that will be your A band. A stands for dark. The I band will be a region where you mostly will have your thin filaments, and as you have your thin filaments, what we see happening is that more light can pass through, which means that it stains on a lighter pattern, thereby creating the I band. So A is for dark, which is mostly going to be thick filaments, and I is going to be for light, which will be mostly your thin filaments. In addition to having the overlap between the thick and the thin filament, also what we're going to utilize, or what we're going to notice within the sarcomere, is that there will be different structural proteins that will help us anchor the thick and the thin filament. So before we start looking at some illustrations, I want to go ahead and point out nebulin and titin. Nebulin will run along the length of the thin filament and will be used not only as a way to determine its overall length, but also as a stabilizing mechanism of your thin/actin filament. Titin, on the other hand, will act almost like a little elastic container that will allow the thick filament, so the filament that's made out of myosin, to be centered and anchored into the sarcomere. And it's going to attach the thick filament to the Z disc. Remember, that's the border. So from one Z disc to the next, that is a sarcomere. And anchoring the thick filament onto the Z disc will be the titin protein. So let's put all these different concepts in an illustrated example, okay? So above over here, we can see our muscle composition within the microscope. And what we can see is that this darkened line right here, this is going to be our Z disc. Okay, and you can see what they've done is here is it under the microscope, and right over here is our illustrated example that might look a little bit more familiar to you at this point. So we have our Z disc to Z discs. And notice how they have bracketed to indicate that that's your sarcomere. So from one Z disc to the next, that is going to be the contractile unit. And within the contractile unit, you're going to notice your thin filament, right? And you're going to notice your thick filament. And you're going to notice that they're in parallel format, one after the other. And that's what's creating our striations. And we can also see that the areas where the thick filament is mostly centered is going to correspond to the darkest region on the top picture. And that is going to correspond with our A band. Whereas the area where the thin filament is centered, those are going to be corresponded with our light or I bands. Okay, so that's the dark and the light that we were chit-chatting about before. We see our Z disc going from side to side. And then another thing that I want to point out to you is if we take a closer look at our illustrated example, we're going to go ahead and we're going to see some of the proteins that we're going to use for structural support, including the fact that we have our titin right over here, which is allowing the anchoring of your thick filament over to your Z disc. You can see the nebulin that is running the length of the thin filament. And then some of you might remember from our previous discussion that we talked about CAP-Z and the tropomodulin. Both of those will also play a role in anchoring and protecting the thin filament. Here is another example of how we can start trying to visualize how all these different components that we're chit-chatting about will work together. We're looking at more of a three-dimensional structure of the sarcomere. So this is what you would find if you were to take a sample of a muscle and you were to zoom in, go over to the myocytes, the individual muscle cells, and just keep zooming in, and you would eventually get to your overall sarcomeres, your contractile units. And as you continue to zoom in, that is then where you would see your thin and your thick filament interacting with each other. Your thin filament, once again, made out of actin, the thick filament being full of the myosin with the heads that are trying to make their cross bridges and their power stroke. Now, usually what we see happening in skeletal muscle is that's a 6-to-1 ratio. Usually you have about 6 thin filaments that will surround one myosin, and that basically allows plenty of opportunity for the myosin head to, when activated, make physical contact with any of the actin active sites. And one thing that you want to keep in mind is that part of homeostasis entails maintaining what we call muscle tone. And muscle tone is basically the resting position that we have to generate for ourselves in a way that we determine what at rest will be the amount of overlap that we have between the thin and the thick filament. And you want to have overlap in the sense that it's going to be easy for you to make a cross bridge once you activate the muscle to start contracting, and that you also have enough room for the thin and the thick filaments to slide past each other, because as they're sliding, that's when you're physically contracting the muscle. So on this little graph right here, it's showing you the length-force relationship, and it's basically trying to show you that you want to go ahead and at homeostasis have this optimal length-force relationship where you can still easily make your cross bridges, but you still have plenty of space to do the power stroking, so the overlapping and the movement. Now, if you go to either extremes, you run into some issues. So for instance, if you at rest have already overextenuated the amount of overlap between the thick and the thin filaments, then what we see happening is when the muscle wants to contract, you'll be able to form your cross bridges. It'll be easy for the myosin head to make contact with the actin. However, there will be no room for them to slide because they've already been maximally compressed. So the muscle won't be able to contract any further. The opposite scenario is if we come to the right-hand side of the graph, and that is that at rest that we overly stretch out the thin and the thick filaments. And what this would then mean is that when the muscle needs to contract, it will have a very difficult time making its initial cross bridges because there's such a low amount of overlap that's occurring between the thick and the thin filament. And if you can't make your cross bridges, that means that you can't do your power stroking, which means that the muscle can't generate the power that you're looking for it to do. An example of this would be, for instance, you might have heard that if you are lifting a heavy item, that you should bend through your knees, not through your back. And the reason people say that is because you have this really long muscle that runs by the base of the skull all the way down the back and connects to the sacrum, the little bone that your little butt bone, your little seating bone. I don't know why I called it a butt bone, but either way. What we see happening is that this muscle, it's called the erector spinae muscle. If you were to bend through your back to lift up a heavy box, what happens is that the erector spinae starts overstretching itself because you're elongating your back as you're hunching over and you're bending. And this then creates a scenario that's very similar to the overstretching that we see happening in our little diagram. And this then means that by the time that you are hunched over and you're trying to lift up that box, the muscle in the erector spinae has already been fully extending itself and it cannot make any cross bridges. So if you now force it, what we see happening is that you cannot make your physical contact, you cannot generate your power stroke, and you cannot generate that energy that you require to lift the heavy box. And this all correlates to you noticing that your back will start to hurt and you're not able to generate the box or lift up the box. On the other hand, if you bend through your knees, then we can use our muscles, for instance, from our gastrocnemius, from our quadriceps, and what we see happening is that these are all huge, bulky muscles that at rest have this perfect amount of overlap so that it makes it easier that when you're in position, you can go ahead and stimulate the formation of the cross bridges and generate enough mechanical energy that you can lift that box without even noticing any issues with the body. Here is also a look at when we take our sarcomere, how exactly is it going to fit together into what we see our muscles look like, right? So we have a little example right over here where we have our skeletal muscle and we have our cardiac muscle. And what we see happening is that the muscles will often take the sarcomere and they'll align them in a linear fashion. And as they're aligning them, they're going to start generating myofibrils. So if you see the term myofibrils, you're looking at nothing but just an elongate structure that is filled with sarcomeres lined up one next to each other. And the more myofibrils you generate, the more they're organized in a parallel format, the more force you're able to generate. So for instance, when you take a look at the muscles that make up your eyelids, they have very minimum amount of myofibrils, which means that you can still contract and relax, but it's going to be a very delicate type of movement. Whereas if we take a look at the muscles that are, for instance, in our biceps or even in our calves or gastrocnemius, we see that we have massive amounts of myofibrils located in those bulky muscles. And the more we bulk them up, the more pressure and the more energy we can generate, and the more work can be done when we force the muscles to contract. Now, I do want to point out something to you, and that is that one of the unique features of a muscle cell is that it's able to be electrically stimulated and to get a response. And part of that excitability comes from the fact that we're able to generate an action potential. Now, in a little bit, we're going to take a look at exactly what is required to start off the sliding filament theory. By now, hopefully, we are familiar with the different components, the thick and the thin filament. We are somewhat getting comfortable with the fact of what the difference is between a cross bridge and a power stroke. So now what we need to do is what actually is going to regulate this contraction, what is going to initiate. And it turns out that it will all start in the plasma membrane of the muscle. Now, a muscle plasma membrane is called a sarcolemma. And what we need to see is that we need to get that sarcolemma to get excited. And when it gets excited, that means that it's going to depolarize. And depolarization basically means that you're going to create a higher level of positivity in the membrane voltage, and the cell will respond by generating action potentials. And as this excitement initiates on the outside surface of the sarcolemma, we see that it will go ahead and propagate and eventually work its way to the inner workings of the myocyte of the muscle cell. And when it does that, that is then when it starts initiating the interaction between the thick and the thin filament. In order to assist with that, we are also going to need to have high levels of calcium inside the cell. And I will go ahead and explain how all these different parts fit together. Just give me a few seconds. But for now, I just want you to know that the excitement starts on the sarcolemma, and then it has to penetrate into the myocyte, into the muscle cell, and it needs to get lots of calcium to flood the internal workings of the cell. And if we have our calcium, and if we have our depolarization on the sarcolemma, that is then when we can go ahead and start initiating our cross bridges and our power stroking. And when we're done contracting, we see that we need to just reverse all those steps, which means that we need to lower the calcium that's inside the cell, and we need to go ahead and repolarize. Repolarize means that we are stopping our action potentials and we are returning back to the resting membrane potential of the muscle cell. And like I said, we're going to have lots of examples as we peruse through our chapters, through our PowerPoints, so please bear with me. For now, depolarization, intracellular calcium equals contraction, and if you want to relax, we're going to have to get rid of that excess calcium and we're going to have to repolarize. Okay, so let's go ahead and take a look exactly as to why calcium plays a role in initiating the sliding filament theory. Now, as you may recall, at the end of our previous part, I discussed the fact that besides the thin and the thick filament, you also have a troponin and tropomyosin complex that's going to be extremely important to the formation of cross bridges. And the troponin and tropomyosin complex, what they'll do is they'll be able to regulate exactly when physical contact can be made between the myosin head and the binding or active sites on the actin. So go ahead and take a look at this illustration, and what it's showing you in red are going to be your actin molecules, so that's your thin filament. And what we see happening is that the binding sites of the actin, here in black, are covered by this almost like purple little filamentous structure. This purple linear structure, that is tropomyosin. okay? And tropomyosin is covering the binding sites on the actin, and the way we're going to be able to move tropomyosin off those binding sites is by this nobule structure that you see right here, and this is troponin. So as you can see, it's a complex, troponin and tropomyosin. They work together. Troponin is the one that will be able to move tropomyosin, thereby exposing the active sites on actin. So what I'm trying to get you guys to visualize at this point is that if you want your muscle to contract, we need to have clear access for the myosin heads to bind to the actin sites. If the muscle is relaxed, then we don't want to have interaction. So at that point, the tropomyosin covers the actin site so that the myosin cannot bind to it at all. So the question now is, what is going to determine when tropomyosin is on the actin site versus when it's off it? We already know that troponin is the one controlling it and moving it, but what signals the movement of the tropomyosin on and off the binding site? And I hope some of you went, calcium, and you're completely correct in that. This is the main reason why calcium is so important for muscle contraction, because when the muscle cell becomes excited and it starts releasing calcium, that calcium will travel over to the troponin and will bind with the TNC region, and the binding of the calcium will cause a conformational change that will allow the troponin to move the tropomyosin away from the binding site, thereby clearing the way for cross bridges to form. Let's go ahead and take a look at more details, information about why the calcium is so important for controlling the formation of the cross bridges and the power stroking. So over here it says that at rest, so when the muscle is relaxed, normally the amount of calcium in the cytoplasm is low. And this allows the troponin and tropomyosin complex to fully cover the actin myosin head. So there's no formation of cross bridges, no power stroking. When the muscle starts to get excited, meaning that there's action potentials occurring on the sarcolemma, when those action potentials propagate their way into the inside of the cell, we see that one of the effects will be that there will be an increase in the concentration of cytoplasmic content. And as the cytoplasmic calcium starts to increase, we see that those calcium molecules will quickly bind to the TNC binding site on troponin. And because it's able to recognize the calcium, it's going to go ahead and cause some conformational changes within the troponin. You know, that's the knob format. One of them is that it's going to go over to the TNI section, which is another receptor part on the tropin. And that is going to loosen up the interaction it's having with the actin. We're also going to see that it's going to signal over to the TNT section, and the TNT section is the part that is able to bind to the tropomyosin, and that will cause the tropomyosin to slide out of the way, and as it's sliding out of the way, it's exposing the binding sites, now freely allowing the myosin head to bind to the actin, aka making your cross bridges. And this will continue as long as you have high levels of calcium. When the cell decides that it's time to relax, to stop the contraction, then part of what we see happening is as the action potential goes away, so does the calcium. And as the calcium is being pumped back, we see that once again, the troponin and tropomyosin will go ahead and slide over those binding sites, thereby eliminating any additional cross bridges from forming. So here we have our picture that we've seen before, but once again, I just wanted to remind you that your troponin is the knob-like format, and as you can see, it has different receptors on it. It has the TNZ where the calcium is going to bind. You have the TNI for the inhibition of the actin and the myosin binding. This is going to lessen when you want to contract the muscle. And then you have your TNT, which is going to be the protein section that can directly communicate with tropomyosin. And it will signal the tropomyosin to slide out of the way, thereby exposing the binding sites. We can also take a look at it from this aspect. So over here, we have our myosin head of the thick filament. We have our thin filament right over here with the actin, and we see that what's happening is that the tropomyosin here in purple is covering the binding site. And the only thing that's going to move it out of the way so you can make your cross bridges is going to be the appearance of calcium. Notice how calcium is indicated by these small little green circles. So the minute the calcium binds, it causes the troponin to move the tropomyosin out of the way, thereby allowing for physical contact to occur. Okay, so this is what it looks like when the muscle is actively contracting, and this is what it looks like when the muscle is relaxed, meaning that the tropomyosin covers the binding site because you are not generating a depolarization or action potential on the sarcolemma, and you're definitely not increasing your calcium concentration on the inside of the cell. Another thing that I want to discuss with you guys is that since skeletal muscle and cardiac muscle both fall in the category of striated muscle, we should definitely take a look if there's going to be any differences when it comes to generating excitation, so your action potential, and coupling that to the internal workings of the cell. So part of what I want to do for the next few slides is let's go ahead and do a comparison between our skeletal muscle and our cardiac muscle, and we're going to tackle 4 differences that we see. We're going to take a look at the time course, so how long does that action potential stay, and how does that correlate with the contraction force that we see. We're going to take a look at what initially causes the depolarization, so the excitement of the membrane to occur. How exactly can we take that action potential and spread it across the sarcolemma and into the myofibrils? And last but not least, where exactly is all this calcium coming in that's going to be flooding in and increasing if we want our muscles to contract? All right, so let's start off with our action potential. I know I've mentioned this word a few times before, and I definitely want to make sure that we're all on the same page and we have a solid understanding of what's happening, because action potentials are very important for our muscle cells as well as for other excitable cells, like for instance your neurons, your nervous tissue cells. So when you generate an action potential, that basically means that you excite a membrane to the point that you get a response. And in our case, the response to the action potential will be that the skeletal and the cardiac muscle will contract. In order for you to generate an action potential, you're first going to have to stimulate the sarcolemma or the plasma membrane. And in order to do that, you're going to do this thing called depolarization. And depolarization basically means that if I'm looking at my chart right here, I see I have a membrane potential. So I have a charge that runs through my membrane, and I have a base level where I start, right? So this is where I started right here. This is called my resting membrane potential. So when the muscle is not being stimulated, when you don't want it to contract, it just kind of sits there, resting at its resting membrane potential. When you want to get the muscle excited because you want to contract, then the first thing we're going to have to do is we're going to have to increase our membrane potential. And when you increase your membrane potential, that basically means that you make the muscle membrane more positive. And that is called depolarization, because you're increasing the membrane voltage. You're making it more positive. And the way you do that is for sodium to come into the cell. So sodium will enter the cell membrane, and as a response, it will depolarize. It will become more positive, and as it's depolarizing, it's becoming excited up to the point that you reach an action potential. And the action potential will be assisted by the fact that along the way, you will also open your calcium channels. And as you are going through your action potential, what we see happening, and this is highlighted in the blue line in our graph, the muscle will respond by contracting. So your action potential will eventually cause your muscle to contract, and it will contract as long as you have an action potential. Now, there will come a time where it's time to kind of go back to your resting membrane potential. So what we see happening then is that we're going to close off our sodium channels, and we're going to switch over to potassium. And we're going to get potassium to leave the cell. And as potassium is leaving the cell, we see that the membrane potential goes back down to where we originally were. And this is called repolarizing. So when you do repolarization, that means that you lower your membrane voltage. That means that you are relaxing or you're calming down the cell membrane. You're going back to your resting membrane potential where your overall base levels were. And this is done by allowing potassium to leave the cell. So if you want to excite, you do depolarization and you allow sodium in. And if you want to relax, you do repolarization, as in you're resetting the cell and you allow potassium out of it. Okay? Now, one thing that we see happening over here is that the overall length of time of action potential is completely different in skeletal muscle than it is in cardiac muscle. In fact, go ahead and take a look at your graph. And let me get rid of this little box right here. Makes it a little easier for us to look at. And we want to take a look at the overall time spent on action potential. And hopefully a lot of you, or many of you, will agree that when you look at your skeletal muscle, you have a very quick action potential. It goes up and it comes right back down. And what this happens with the quick action potential means that you generate what we call a twitch, a quick contraction. And it turns out that it's okay for skeletal muscles to twitch because it does it so quickly that to the naked eye, those twitches fuse together and create a smooth contraction and relaxation. So if you were to look at your fingers right now and I were to tell you, go ahead and bend your fingers and contract them, you might notice that it's a smooth movement, but on a microscopic level, what we would see is the accumulation of thousands of twitches, because the action potential goes so quickly up and down. On the other hand, if you take a look at your graph for cardiac muscle, notice how the action potential has a very slow plateau. And this slow plateau is mostly due to the fact that the sodium channels will stay open a little bit longer, as well as the calcium channels will stay open a little bit longer and are more sensitive in cardiac muscle, allowing for the action potential to have a longer time limit. And this then means that when the cardiac muscle contracts, it doesn't twitch. It does a slow, strong contraction followed by a relaxation. We don't want our cardiac muscle twitching away because its main purpose is to create forceful contractions that allow the heart to pump, thereby generating enough pressure and force for blood to be distributed throughout the rest of our body. Another difference that we see between skeletal and cardiac muscle is what exactly causes the cell to start initiating depolarization. So how does the cell know that you want it to contract? And we have two options. We can go ahead and take a look at a myogenic versus a neurogenic initiation. Myogenic means that it begins within the muscle. This usually means that it's a spontaneous reaction. Usually that means that you have a set of cells that will initiate the excitement and that are able to depolarize very quickly, and we call those cells pacemaker cells. In fact, when we look at the heart, our cardiac muscle, it does myogenic initiation. And the reason for that is because our individual heart cells are able to spontaneously contract and relax because they're very excitable. However, what we see is in order to generate a very strong heartbeat, we're going to have almost like leaders of the pack, cells that initiate this contraction. Those cells are the pacemaker cells. Some of you might know them as the sinoatrium node, the SA node. It's a collection of cells that sits in the right atrium, the top chamber of the heart. And these cells are the first ones to fire off their depolarization and induce their action potential. And they do so because they have these F channels. F channels actually stands for funny channels, and it gets its name because it goes ahead and causes an imbalance of sodium and potassium, and it's very quick to initiate the calcium release as well as the sodium release. So what we see is that these cells, as soon as they repolarize, they will depolarize again, and it will spontaneously cause action potentials to build one on top of each other. And this is why, on average, the heart will collectively beat every 0.8 seconds, because their cells are so excited that they are continuously going through the depolarization, action potential, repolarization stage. Your skeletal muscles, on the other hand, have a completely different story. They rely on neurogenic control. And neurogenic control means that the muscle cell itself cannot initiate anything without the message or control from our motor nerves. So we rely on our nervous system, and what we can do is we can go over to our illustration on the right-hand side, and it's showing you a motor neuron. It's showing you the axon, the end part of your neuron cell, and as you can see, it's going to go ahead and make a physical connection. And you know what, it shouldn't say physical connection. It's going to get very close because it doesn't actually physically connect. It's going to hover its synaptic knob, which is the end part of the axon for the neuron. It's going to hover it over the sarcolemma. So the plasma membrane of the skeletal muscle, we call this a neuromuscular junction. And what we see happening is, if you want your skeletal muscle to contract, we are going to need a nerve signal to come down the axon, release acetylcholine, and only when it releases its excitement and its neurotransmitter will the muscle cell respond by initiating depolarization and initiating an action potential. This is one of the reasons why, for instance, when someone gets into an accident and there's damage done to the spinal cord, we can often see that some paralysis might occur within the skeletal muscle. Because if there's damage to the spinal cord and you cut off the communication between the motor neuron and the skeletal muscle, you will not be able to stimulate the muscle to contract and relax. And that's because skeletal muscle is under neurogenic control. It solely relies on the nerves to tell it when it's time to contract and when it's time to relax. Another difference that we see in cardiac and skeletal muscle is its composition of T tubules and SR regions. SR stands for Sarcoplasmic Reticulum. Now, the reason you need these two items is because it's going to help you extend the excitement that you see on the sarcolemma, the outside of the muscle cell, to the inside where the thin and the thick filaments are. So go ahead and take a look at your illustration, and we can see we have our little sarcolemma pictured in both examples. So that's the plasma membrane of the muscle cell. And as I mentioned to you many times before, is that you want to go ahead and generate an action potential on the sarcolemma first, get that membrane nice and excited. And now what we see happening is that the excitement starts to spread across the surface, and it will look for these green structures called T tubules, transverse tubules. Transverse tubules are best described as invaginations, but I like to think of them as a little tunnel, and they are going to allow you to take the excitement that's on the outside of the sarcolemma and penetrate to the inside of the cell. See how it expands to the internal workings of your myocyte? And as it penetrates to the inside of the cell, it's able to excite all that internal workings. And more importantly, it's going to stimulate the sarcoplasmic reticulum, indicated by all the purple regions, to release calcium. So the function of the SR region, the sarcoplasmic reticulum, is to store calcium. So whenever the muscle gets excited, it has access to calcium that needs to be released into the intracellular or cytoplasmic components of it. Now, usually the T tubules and the sarcoplasmic reticulums will create what we call a triad region. And the triad region usually means that you have one T tubule in the middle and a sarcoplasmic reticulum on either end. So one T tubule and then two sarcoplasmic reticulums that are surrounding it, and collectively that makes a triad. Now, what are the differences? Well, in skeletal muscle, we tend to see that they have more extensive T-tubules and also a lot more sarcoplasmic reticulum, and it's not as developed in our skeletal muscle, in our cardiac muscle. And this obviously can correlate to the fact that our skeletal muscle tends to be larger and of course is on that twitching fest, so it responds a lot quicker than your cardiac muscle. Now, let's go ahead and take a look at one more difference, and that's going to be exactly how do we get calcium to the inside of the myocytes, into their cytoplasm if we want the muscle to contract, and also how do we get rid of it if we want the muscle to relax. So in order to do that, we're going to rely on different channels to allow the calcium either to enter through the cytoplasm or, to allow the calcium to enter into the cytoplasm through the membrane of the cell or from the sarcoplasmic reticulum, the SR region, because we know that's where we store our calcium. And also we're going to need to rely on different ATP transporters to kind of pump out the calcium if we want to lower the calcium rate in the case of wanting to relax the muscle. So let me go ahead and show you the names of these different channels and transporters. And then I do have an illustration on the next picture that makes it a little bit easier to try to visualize the concept that I'm trying to come across. Okay, so two divisions on this PowerPoint. The first one is going to concentrate on how do we get calcium into the cytoplasm. So how do we increase the concentration of calcium on the inside? And this is obviously done when we want our muscles to contract. And we see that we can have some calcium channels that will allow calcium to go from the outside to the inside of the cell through the cell membrane itself. Or we can get the calcium to be released from the sarcoplasmic reticulum, so it can also increase its content intracellular. If we're looking at the cell membrane, then we're looking at our dihydropyridine receptor, DHPR. That's going to be the channel that's going to regulate movement from calcium through the cell membrane. For our SR membrane, we're going to look at the ryodine receptor. This is going to be the channel that we're going to rely for calcium to enter into the cytoplasmic content. So we're all doing this in an effort to increase calcium so we can get our muscles to contract. If we want to remove the calcium because it's time for the muscle to relax, then what we see happening is that we're only going to use active transporters. So this is going to require a lot of ATP to get this done and to remove the calcium. In the cell membrane, you will have two transporters. You will have the calcium ATPase and the sodium calcium exchanger. These two transporters will help you pump out the calcium from the cytoplasm. And from the SR region, the sarcoplasmic region, we're also going to notice that we have our SERCA transporter, which is another example of a calcium ATPase. Now, the function of these three, the calcium ATPase, the sodium calcium exchanger, and the SERCA, is all to remove the calcium from the cytoplasm so that the muscle can go ahead and relax. All right, so let's take a look at this from an illustrative aspect. So on this picture right here, we can see that we have our sarcolemma. So this is our cell membrane. This is the outside of the cell. And this is going to be the inside of the cell. So this is where we have our cytoplasmic content. And we can see we have our SR region right over here. And then now let's take a look at the different channels. So let's start off in the cell membrane, okay? So go ahead and take a look at your receptors that are pictured on the cell membrane. So in this section right here, and go ahead and identify the ones that will allow calcium to come into the cell and those that will allow calcium to come out of the cell. And I'll help you along the way. So follow the arrow. So the first one I want to look at is this one right here, labeled DHPR. That is going to be the channel that I'm going to use to pump calcium into my myocyte. So to increase the cytoplasmic concentration of calcium. I'm doing this when I want my muscles to contract. On the other hand, if I look on either side of my DHR, I'm going to notice that I have my sodium calcium exchanger. This is going to allow me to take my calcium and pump it outside in exchange for a sodium. And I also have my calcium ACPase. Both of these will function in the role to remove the calcium from the inside of the cell and move it outwards. And these two membranes or these two channels will be utilized when it's time for the muscle to relax and restore itself back. So we no longer want to have our cross bridges and we no longer want to have our power stroke. If you now come over to this Sarcoplasmic Reticulum, your SR region, where you're storing lots of calcium, what we see happening is that if we want the calcium to be released from the SR region and go to the inside of the cell, we're going to rely on our ryanodine receptor. Okay, see how the arrow pumps the calcium out of the SR and into the cytoplasm? Whereas if we want the calcium to come back into storage and we want to pump it out of the cytoplasm into the SR region, then we're going to rely on our ATPase titled Circa. All right, now please notice that you're going to have to require ATP for all of the transporters that are going to help you remove calcium from the cytoplasm. All right, please make sure that for your quizzes as well as for your exam that you know the difference between your DHPR, your ryanodine receptor, your calcium ATPases, your sodium calcium exchanger, and you know exactly where they're located. Is it in the membrane? Is it in the sarcoplasmic reticulum? And if it's allowing calcium to come in or if it's allowing the calcium to exit the myocyte. So it turns out that both the skeletal muscle and the cardiac muscle will have the same channels that will allow movement in and out in both the sarcolemma or the cell membrane as well as the sarcoplasmic reticulum. What will change or what will be the difference is exactly what will cause the calcium to be released from the sarcoplasmic reticulum. So what this one tries to explain to you is that as the action potential is being generated on the sarcolemma, on the outside cell membrane, it will go ahead and spread on the outside until it will encounter a T-tubule, and the T-tubule will spread all that excitement and depolarization into the cell. And as it's spreading it, what we see happening is that for both the cardiac as well as the skeletal muscle, the first receptor or channel to respond to the excitement will be the DHPR. And the DHPR channel, as you recall, is located within the sarcolemma, the cell membrane, and it will allow calcium to go from the outside of the cell to the inside of the cell, right? And it turns out that once the DHPR is activated, the next step is to get the sarcoplasmic reticulum to join in on the fun and also release the calcium that they're holding on to. However, the response of the sarcoplasmic reticulum will differ in the cardiac versus the skeletal muscle. So let me tell you what the difference is. In the heart, we're going to see this response called calcium-induced calcium response. And what this means is that the minute that calcium starts flooding into the cytoplasm of the myocyte because of the opening of the DHPR channel, the minute that the calcium concentration becomes elevated, the ryanodine receptor will notice that as well, and it will allow itself to open up, allowing calcium to be released from the sarcoplasmic reticulum. So the presence of calcium causes even more calcium to be released. And in fact, the illustration that you see on the right-hand side right over here is highlighting your cardiac muscle, and it's showing you how calcium causes even more calcium to be released from the sarcoplasmic reticulum. In skeletal muscle, what we see happening is that, yes, just like in cardiac, the DHPR channel is the first one to open up, and it's the first one to allow calcium to flood into the cell. But the sarcoplasmic reticulum and its ryanodine receptor doesn't wait to see if the calcium concentration even increases before it opens up. In fact, what it will do is as soon as it notices that the DHPR receptor goes through a conformational change and is activated, it will automatically respond as well and open up. So it doesn't wait for the concentration of calcium to increase. It goes ahead and responds to the depolarization directly of the DHPR receptor. So in that case, what we have is we have a depolarization induced calcium release. So just because one receptor opened up, the second one opened up as well and was excited. We can go ahead and move on to the next slide to see that in illustrative example. All right, so here we go. We can see that the excitation and the depolarization of the plasma membrane will cause the DHPR membrane to be activated, and as calcium floods in, what we see happening is that due to the fact that you had the change in membrane voltage, it will also cause the release of calcium due to the ryanodine receptor. So in the cardiac muscle, we wait for the calcium concentration to go up. In skeletal muscle, we simply respond by the fact that one channel is opening because of depolarization, so the second channel will respond to that as well. Now that we have sort of tackled depolarization and the movement of calcium into and also out to a little bit of the cell, and obviously by now we're total experts on cross bridges and power stroking, right? I can hear you agreeing with me. Let's go ahead and take a second and just reflect on relaxation, because I keep mentioning relaxation, but I definitely want to make sure that we collectively know what the events are that will cause the muscle to eventually relax. So on your slide right here, I wrote down for you that if you want to have muscle relaxation, we need to have repolarization of the sarcolemma, and repolarization means what? It means that we lower the membrane voltage, and we do so by allowing potassium to come out of the cell. Okay? And as repolarization is initiated, that means that we need to get rid of that excess calcium that's floating in the cytoplasm. And the way we're going to do that is by utilizing ATP transporters in both the sarcolemma and your SR region, the Sarcoplasmic Reticulum. Now, some organisms, not humans, will also utilize another protein called parvalbumin. This is going to be a calcium binding protein that will basically speed up the removal of calcium from the cytoplasmic contents. Humans really don't have access to it, and we don't really need to utilize it. But in certain fish species that have high levels of white muscle that could respond quicker, we do see that the parvalbumin is very effective in getting rid of that excess calcium so that the muscles can fully relax at a quicker rate. Either way, once you get rid of all the calcium that's in the cytoplasm and you either kick it out of the cell or you store it back into the sarcoplasmic reticulum, we see that there's no longer any calcium to interact with the troponin. And that means that the tropomyosin will slide back over the binding sites on the actin, thereby inhibiting the myosin head from making physical contact. So no more cross bridges. And if you can't make your cross bridges and you can't bind your myosin and your actin, then the muscle will ultimately relax, waiting until the next time that it needs to go ahead and contract. It's also important, since we talked about the difference between skeletal muscle and cardiac muscle, to go ahead and take a peek what's happening in smooth muscle. Smooth muscle is obviously also able to contract. However, it has some key differences. One of them is obviously the fact that it doesn't have the striations, and part of that is that it doesn't have a very organized pattern of its thick to its thin filament. They are scattered all over the place, and in fact, what you can do is you can go ahead and take a look at your illustration, and you can see the thin and the thick filaments pictured in irregular patterns. They will anchor through the plasma membrane through an adhesion plaque. There is no mention of a sarcomere because you don't have that level of organization. They don't have T-tubules. They have very minimum sarcoplasmic reticulum. They will often utilize gap junctions to share ions from one cell to the next. And because they don't have that level of organization, when the smooth muscle contracts, instead of having a smooth, overlapping, sliding filament analysis, we get this irregular contraction from all sides. Because as many thin filaments can interact with thick filaments, they'll try to do it and they'll try to slide over each other, creating this irregular, almost like a puffy format as it contracts and it relaxes. It also has a very different mechanism that will control the troponin and the tropomyosin. And part of that comes from this very important statement right here. Smooth muscle does not have troponin. It does have tropomyosin, but it doesn't have troponin. And in fact, what's going to happen is that in smooth muscle, one of the big differences is that it's going to use caldesmon, which is another protein, and this protein will be binding to actin and will be blocking the myosin binding when the muscle is relaxed. Now, before we talk about that a little bit further, I want to point something out at the top of the slide. Over here it says that the contraction of the smooth muscle is regulated by many different things, including nerves, hormones, as well as physical conditions such as stretch. This is a huge difference from skeletal muscle because, as you might recall from our previous slide, your skeletal muscle can only be controlled by neurogenic stimulation, meaning that the nervous signal has to be sent before it can respond. Smooth muscle can also rely on nerves, but it does have alternate options to initiate its contraction. All right, back to our caldesmon. What we see happening is when it's time for the muscle to contract, we do need an increase in calcium. The calcium, however, will form a different pathway. It will first bind with calmodulin, and calmodulin will then go ahead and initiate the caldesmon, and this will allow the removal from the actin of the tropomyosin, thereby allowing your cross bridges to form. Your calmodulin is also important because it will initiate some of the phosphorylation, so it will help with some of the ATP's activity that you have going on in the myosin head. Now, as we finish up our chapter, I do want to go ahead and take a look at some what they call transdifferentiation, meaning what are some unique functions that we come across in different organisms for the use of muscle cells. And when you do transdifferentiation, what you're looking at is that you try to use the cell for what they call a novel function, so for something that you wouldn't expect. Now, in the case of muscles, you would just expect it to be used for contraction, but it turns out that we can also use it to help generate heat as well as electricity. So for instance, on this example right here, you can see that they've drawn a marlin for you. And the reason they've done that is because we see that in the eye of the marlin, they will have muscles that will serve as a heater tissue. And these muscles, we're not as concerned with getting them to contract and relax, but what we do want them to do is we want them to go ahead and respond to excitation by creating a vicious cycle where calcium goes in and out of the sarcoplasmic reticulum. And as the calcium just gets pumped in and out both directions, it will correlate with a high rate of ATP production and consumption. So you can see right over here in the illustration, we see our calcium cycling in and out, and as an end result, we also see our ATP production and utilization. And this response to getting the muscles excuse me, to depolarizing the muscles, this response of activating the calcium and the ATP production, instead of getting the muscles to contract, will actually cause the muscle cells to go ahead and produce a massive amount of heat. And it turns out that that heat will directly correlate with the optical sensory system that is by the eye. So you can see its close relationship to the eye right here. And as the heat interacts with the optical sensory system, it actually allows the marlin and the billfish to have a better visual of the water that they are swimming through. So it actually increases with their ability to see in deep and cold waters. How cool is that? They use the muscles for that. In eels, we see the same exact concept of generating the cycle of calcium and ATP, but instead of generating heat, they use their muscle cells to basically make short electrical currents. And in fact, we like to call those muscle cells electrocytes because of their electric capability. And that is then what gives that electrical discharge on an electric eel. Another difference that we see when we start taking a look at muscles and comparing it across different animal models is that in invertebrate muscles, they have this thing called a graded excitatory postsynaptic potential, EPSP. And the reason it's graded means that when you start stimulated, you can basically summate a stronger response out of the muscles. This is very unique because our muscles are all or none. So once you reach the threshold of stimulation, our muscles have to contract, and they will contract to the maximum. We generate more and more muscle strength by recruiting more and more myofibrils, so muscle fibers, but the individual myocytes either contract or they don't. In invertebrate muscle, on the other hand, what we see happening is that they can have a graded response, meaning that you can start increasing the threshold and increasing the stimulus, and you will start seeing a more amplified result that will keep building until you reach your fully saturation point. You also see in invertebrate muscle something that we absolutely can't do, which is that a nerve signal can actually cause the muscles to hyperpolarize, thereby inhibiting its ability to contract. That is the complete opposite of what our skeletal muscles do. Our nerve signals are meant to actually induce muscle contraction, not inhibit it. Another comparative anatomy aspect for muscles is that of asynchronous insect flight muscles. This one's a pretty cool one. So basically what they want to figure out is what is the limit of how many excitation contraction couplings we can do. So basically, how quickly can we contract the muscle? Is there a limit to how much we can do? And for vertebrae, we see that our limit is about 100 times a second. That's the fastest that some vertebrates can contract their muscle. Okay, and this is based on its limitation to how many cross bridges you could make, how much overlap you have with your overall power stroking. Now, what we see happening is that when we take a look at insects and we take a look at how they use their flight muscles to basically cause their wings to go up and down, we see that they do this thing called asynchronous muscle contraction. And this muscle contraction basically means that it doesn't continuously rely on nerve stimulation. And because of this fact, it's able to alter its calcium sensitivity because it's going to utilize this thing called stretch activation. And due to the fact that it doesn't synchronize with the nerves all the way and it has the stretch activation, it can increase the amount of contractions to anywhere from 250 to 1,000 contractions per second. That is extremely fast. So try to visualize, for instance, a hummingbird. And when you think about how fast it has to whip those wings, that basically corresponds with how many contractions and relaxations it has to do. And obviously it does it a lot faster than we can move any of our muscles. And part of that is due to the fact that it's able to do the stretch activation. And the stretch activation means that it's going to be able to alter how sensitive the individual myofibrils are to calcium. So let's take a look at our bottom part of our slide right here, and it says that your intracellular calcium will remain high the minute that your action potential is induced. So as you can see on my graph right here, we're looking at our insect flight muscles. We have done our depolarization step, and our calcium level has shot up, so we're good to go. But what's going to end up happening is that we don't want the muscle to contract the whole time. We want the muscle to contract and relax, thereby allowing the wings to beat up and down. And since we're not going to be relying on nerve signal the whole time, we're going to utilize that calcium by increasing or decreasing our sensitivity to it. So what we see happening within the muscles of the flight, or of the wings, excuse me, is that the minute that the muscle contracts, it becomes calcium insensitive. So it no longer responds to the high levels of calcium. At that point, the contracted muscle will start to relax, and as it's relaxing, what we see happening, because they have elastic elements within their wings, the minute the muscle relaxes and starts stretching and lowering itself, it will go ahead and switch back on its calcium sensitivity. And as the calcium becomes once again sensitive, or I should say, the muscle once again becomes sensitive to the calcium, the muscle will respond by contracting and the wings go right back up. And once again, we do that all over again. So as the wings go up, it loses its sensitivity to calcium, the wings fall down, and the elastic recoil will go ahead and cause the calcium sensitivity to come back in, and the wings go right back up. And they do this very quickly. And then our last slide is to take a look at the catch muscle. The catch muscle is what you'll come across when you look at items like clams or oysters. And it's basically their ability to kind of hold the shell closed. It's an example of an adductor muscle. And what's so unique about this catch muscle is that it's able to maintain an extended period of contraction without using a lot of energy. In fact, it uses little to no energy. Instead, what it's going to use this protein called twitchin. And twitchin will be regulated by if it goes through phosphorylation or dephosphorylation. So what do I mean with that? So the minute that the shell closes off, it goes ahead and it contracts this catch muscle. And normally what we see happening is that as it's contracting the catch muscle, the muscle is active. And as we've discussed previously with our skeletal muscle, if we want to have that muscle keep contracting, we're going to have to pump lots of ATP into the system, and obviously calcium as well. But in fact, what we see happening with the catch muscle over here of our mollusks is that the cross bridges do not continue to cycle, and that's because the twitchin protein is going to go through its dephosphorylation step the minute the muscle becomes active. And when we see that it starts to phosphorylate again, that will then correspond with the end of the contraction. And as you can see, it takes quite a bit of time before it starts to phosphorylate. And all that time, the contraction is being maintained without the help of calcium and without the help of ATP. So it's all about the twitchin protein and its phosphorylation, dephosphorylation step that is able to maintain the muscles holding on to their physical connection in the cross bridge and not cycle. So you hold on to that one contraction for an extended period of time without spending any energy on it. Very unique setup. All right, I think that's all I have for today. So go ahead and, like you did before, please take your time, look through the video, take your notes. Make sure to reach out to me if you have any questions, comments, or concern. I do want to point out that Chapter 5 is the last lecture that we do before we take our Unit 1 exam. So definitely keep an eye on the class schedule for when your exam becomes available. And like I said, if you have any concerns, please reach out to me before you take the exam, not after, okay? All right, we'll talk soon.